Hello again. We're going to continue our week of connections by talking about a really relevant topic. We're going to talk just a little bit about viruses. Like everything else this week, the point of today is just to give you enough background information to understand a lab that we're going to do tomorrow. We will spend a little more time learning about viruses in more depth. We will also learn more about the immune system later on, actually, at the beginning of second trimester. So today is just kind of an introduction to these topics. To start with, spend a couple minutes taking a look at this interactive. This is just a click and learn. When you click on this link, click the button that says Launch Interactive and just spend a few minutes looking around at some of the different viruses, the shapes, the sizes, the types that exist. So a virus is a really interesting biological thing because it is not exactly a living thing, but it has some of the characteristics of life. A virus is an infectious particle that consists of, it's a pretty simple structure. It has some kind of genetic material. It could be DNA, it could be RNA, packaged in a protein covering. And the coverings are a little different with different kinds of viruses, but that's the basic structure of viruses. They're really, really tiny. They're smaller than ribosomes, which, if you remember, ribosomes are protein-making structures within our cells. So they're even smaller than those. And the genetic material can be different for different types of viruses. It can be double or single-stranded, and it can be DNA or RNA. The outer covering is called a capsid, and it can vary in shape depending on the type of virus, as you saw. Some also have a viral envelope, which is similar to a membrane. So there is some variety in the structure, but overall they're pretty simple. It's some kind of genetic material with a covering. Viruses are a type of thing called an obligate intracellular parasite. What that means is that they can only replicate in a host cell. So obligate means they have to. Intracellular means inside of a cell, and parasite means that they're living off something else. So viruses can only survive if they can use energy and materials and resources from the cells of a living creature. This is one of the main reasons that viruses are generally not considered alive, because they can't reproduce, they cannot produce their own energy without using a host cell. The host range is how we describe the number of species a virus can infect. Some viruses are very specific and only infect one species. Others maybe can infect mammals, but not other types. And others can range across a broader spectrum of living things. And the host range is really identified by the match between the viral proteins and receptor molecules on the cells. Because what viruses do is they use some of the structures on the surfaces of our cells to kind of let themselves into our cells, trick our cells into taking them in, and then they can use our cellular machinery to reproduce themselves. We're going to take just a couple minutes and look specifically at the virus that is on all of our minds and some of the receptor molecules that it uses. So we're going to look at a couple of the figures from that reading and label some things to kind of see how this works. This is a diagram of coronavirus, and you're going to label a couple different parts. Now we're going to review a little bit of cell membrane structure so that we can make some connections here. Then finally, we're going to try and tie all of this together. In this diagram, this HSPG is that carbohydrate that was being discussed in that article. And the ACE2 is the protein receptor. So this is a protein attached to a carbohydrate that is necessary. The SARS-CoV-2 is the virus that causes COVID-19. It has to attach to these carbohydrates on the outside to help it reach that protein receptor. And the protein receptor is what helps it get taken into the cell. So think about what you read in the article and how the treatments they're suggesting might help to prevent infection from COVID-19. So now we're going to go back to just talking about viruses a little more generally. We're going to watch a short video that shows how an influenza virus, so this is a different type of virus, this is not a coronavirus, it's a flu virus, but how it infects a cell and replicates itself. And there are a lot of similarities here to what you just read about. This is explained pretty simply. It's made for the general public. But as you watch it, try and think about what you see in here and what vocabulary and structures you can identify. And we'll pause and talk about some of those as we go.
So let's say that this guy has the flu, could be any flu, and here's a droplet from his sneeze containing, if we move in and take a really close look, you see each one of those little purple things is a virus. And there are a lot of viruses floating through the air, some of which go inevitably up this unfortunate man's nose. How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay, I am talking with medical illustrator David Belinsky, who designed this video for Xyrus, a research company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah.
These keys, of course, if this was the COVID-19 virus that you were just looking at, these are those spike proteins. So these are proteins on the outside of this virus. They're using an analogy of a key and a lock.
This is a key, this is a key, this is a key. Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the...
So those little purpley stick-uppy things, as he called them, are the receptor proteins that can also be attached to those carbohydrate chains that help the virus to kind of latch on to the outside of a cell.
Surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus.
So those virus recipes, as he calls them, of course, are the genetic material of that virus. And this big blue thing with the holes in it is the nucleus of this cell. So the virus is now inside, and it's going to kind of take over the cell's machinery to make copies of itself.
So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus, where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface, where they get new keys, and then boom, here they come. This is an eruption of virus after virus after virus after only one virus entered the cell. But how many came out? Well, millions. So if one virus can produce 1 million babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1 million viruses is just a drop in the bucket. We can have 100 trillion cells. And anyway, remember, you do have your own immune system, which when it sees a virus usually kills it. So while the virus does multiply fast, with any luck your immune system will work just a little faster. So, yes, all viruses want to spread. That's what they do. But most of the time, we do keep them in check. Most of the time.
All right. So we're now going to shift over and talk a little bit about the immune system. So this is something, like I said, that we are going to come back to in much more detail later. So if this looks a little complicated and overwhelming, don't worry. I'll move myself out of the way here. But we're just going to talk kind of about the basics. So first of all, this process starts when your body is exposed to something called an antigen. That's just any substance that triggers an immune response. It could be a molecule on a bacteria. It could be those proteins on the outside of a virus. It could be a fungus. It could be a lot of different things. That antigen triggers a whole cascade of events that allow your body to fight off that invader. There are lots of different types of cells involved in this process. But in that process, they also create what are called memory cells that remember that particular invader in the future, so that if you get infected again, your body can respond more quickly. And most importantly, for what we're going to be talking about here, part of that immune response is that your cell also creates antibodies, which are proteins. You can see them down there in the bottom left. They're kind of Y-shaped proteins that recognize the pathogen that is in your bloodstream and will kind of bind to it so that it's covered up and can no longer enter your cells. So it kind of disables that antigen. And those antibodies will stick around in your bloodstream for a while, even after you've been infected. And so after you encounter a pathogen or an antigen for the first time, it takes your body a while to kind of gear up and make the antibodies. And once it has enough antibodies, you start feeling better. But because those memory cells and some of those antibodies stay in your body, the next time if you encounter the same thing, your immune system responds much more quickly. So when we talk about testing for a disease like COVID, there are kind of two ways to do it. We can either test for the virus itself. That would be testing for the pathogen to see if you are actually infected at this current moment. But the other type of testing you might have heard about is antibody testing, which basically looks to see, has your body activated that immune response and produced antibodies that are still floating around in your bloodstream, which means you were exposed to this or were infected. You may or may not be sick anymore, but you still have those antibodies in your bloodstream. So you might have heard about both of those. The lab that we are going to be doing tomorrow is called an ELISA test. This actually is the exact same procedure that they use to test for COVID-19 antibodies, one of them anyway. In our lab, everything will be simulated. We are not actually testing for COVID-19 antibodies, but it's the same technique and the same procedure. So this is called an ELISA. That stands for Enzyme-Linked Immunosorbent Assay, which is very fancy. Enzyme-linked just means they use an enzyme to help show the results. You'll see that in a little bit. Immunosorbent means that something from the immune system is being linked on or attached to something else. And an assay is just a type of a test. The procedure that we're going to use here kind of looks like this. First, the antigen in this case, if we are using the analogy of COVID-19 or the scenario of COVID-19, the antigen would be those proteins from the outside of a COVID-19 virus are attached to a plastic assay plate and bond to the surface. So this A down here is the antigen. Then we add the sample, because our question here really is, are there antibodies in this sample? So what we're trying to test for are these green antibodies right here, because if this antibody is present, it will bond to that COVID-19 protein, if that's what we are testing for. So this is what we want to know, whether this is there or not. If that, oops, sorry, I don't know what just happened right there. Here we go. If that antibody is present, Then when we add the next step, the secondary antibody was made in a laboratory with an enzyme attached to it, and it's made to bond to the primary antibody, that antibody against COVID-19. So if that's there, the secondary antibody will bond to the one we put in first. And then since it has that enzyme attached to it, that enzyme catalyzes a reaction that takes this substrate molecule and changes the color. And so it gives a product that has a color that we can see. So if it changes color, that means this antibody was there because it allowed this to link and this to link. And if that antibody is not here, if this person does not have COVID antibodies, then when we put this one in, there will be nothing for it attached to, so it'll just get washed out. And then there will be no enzyme, and so there won't be anything for the substrate to link onto, so the color change won't happen. So that's kind of how we are going to be going through and testing for this. This ELISA procedure is used for a lot of things. It was actually first used for testing for HIV, and it's used for other viruses as well. It can be also used to test for food allergens or genetically modified food substances, things like that. And of course, it can be used for detecting viruses. This, remember, is not testing for active infections. There's a different technique that was used for this. An ELISA test like we're doing here tests for past infections to see if you have been exposed to this previously and have the antibodies in your bloodstream. The benefits of this are to figure out whether someone has some level of immunity and figure out how long that lasts if we test people repeatedly over time, which helps us to understand the antibody response. This is something that scientists are still doing a lot of research on, specifically for COVID-19, so that they can start developing treatments and vaccines. Obviously, there's a lot of research being done in this area right now. ELISAs do have some limitations, though. It's possible to get false positive and false negative results, and it takes some time. False positives can happen if perhaps this test is detecting another coronavirus. Coronaviruses are a big family of viruses, and COVID-19 is just one of them. So you can get some false positives. You can also get false negatives, and this is something that's really important to remember when you talk about people being tested, because this is also true of tests for the virus itself. If these tests are taken too soon, remember, it takes your body a while to activate that immune response. And so if you were to take a test like this, say the day after you were next to somebody with COVID, you'd probably get a negative result, even though you have it in your body, but your body hasn't had time to kind of replicate the virus enough and ramp up the defenses enough for anything to be detected in a test. So just because you have a negative test doesn't always mean that you don't have that virus or didn't have it in the past. And this does take a little bit longer because that immune system takes a little bit of time to respond. So there are limitations to this. But it is still a widely used test. This is a chart that I found that was published in August of 2020 that lists several of the tests. These are all antibody tests that were authorized for use for COVID-19. And you can see here at the bottom, 5 of them are these ELISA tests using this exact same procedure that we're going to be using in class. So tomorrow, we will do this lab to see who has antibodies for COVID-19. Like I said, not really simulated, but same procedure. Couple of reminders. You have a couple of things to be working on. We're coming up on the end of the trimester, so make sure you're checking the gradebook as well. And also, those of you at home, if you are interested in joining class virtually to see this lab tomorrow, shoot me an email or arrange it with a friend of yours who is in House B and will be in class tomorrow. And then please just let me know who you're going to be working with. So we will see you tomorrow for our ELISA lab. Have a great day.